DNA, or deoxyribonucleic acid, is a negatively charged molecule that contains the code to control the genetic makeup of humans and almost all other organisms. The information in DNA is stored as a code made up of 4 nitrogenous bases: adenine, guanine, thymine, and cytosine. These bases form nucleic acids and are the building blocks of DNA. Nitrogenous bases are composed of carbon and nitrogenous rings. The number of rings a base has determines whether it's classified as a purine or pyrimidine. Adenine and guanine have 2 nitrogen rings, making them purines, whereas thymine and cytosine have 1 ring, making them pyrimidines. The purines on one strand of DNA form hydrogen bonds with the corresponding pyrimidines on the opposite strand of DNA, called base pairings. Base pairing rules determine that adenine will pair with thymine and guanine will pair with cytosine. The order or sequence of these bases determines the information available for building and maintaining an organism. Each base is also attached to a sugar molecule and a phosphate molecule. Together, a base, sugar, and phosphate are called a nucleotide. Nucleotides are arranged in 2 long strands that form a spiral called a double helix. The long strand runs in opposite directions, one end being called 5' and the other end the 3'. 5' indicates the 5 carbon on a deoxyribose sugar, which is attached to a phosphate, whereas the 3 carbon sugar attaches to a hydroxyl group. The structure of the double helix is somewhat like a ladder, with the base pairs forming the ladder's rungs and the sugar and phosphate molecules forming the vertical side pieces of the ladder. An important function of DNA is its ability to replicate itself. The weak hydrogen bonds which join base pairings can easily be separated, forming 2 individual strands. With the assistance of DNA polymerase, the separated strand acts like a template and creates an identical strand of itself. Without such a vital process, the human genome would eradicate itself over time via mitosis.